Somebody said yes, definitely, right? Good. All right. Well, we are set to... Oh, come on, guys. Where did we go there? We already saw all that, didn't we? Well, let's see how we go there. All right, there's where I wanted to start. So last time I got kind of hurried at the end, as oftentimes happens with me, talking about, why that's a little too loud, talking about the weak acids. Okay, so I want to go back over that a little bit and make sure we get this hammered home. So what you see on the screen is a titration of a weak acid, and we see it between, okay, two different forms. Titrations will happen between different forms of acids. We see in this case carbonic acid, H2CO3, ionizing and becoming bicarbonate, HCO3-. We see on the y-axis a plot of pH. We see on the x-axis a plot of equivalence of hydroxide added. Okay? Very important thing that's going to happen in a titration, obviously, right? I've marked various places on the graph where you see on the lower left, I've marked H2CO3, meaning that at that place on the graph, the predominant molecule that's there is H2CO3, carbonic acid. As I keep adding hydroxide ions, the pH increases, and you see the pH increases relatively rapidly. That is, that orange thing is going up fairly rapidly, and then it starts to level off. And that leveling off, as I indicated last time, is what's happening when a buffer kicks in. A buffer is resisting the change in the pH. And we see that for this particular buffer, that buffering starts to happen at about pH 5.37, thereabouts. And it stays relatively flat until we get up to about pH 7.37. Well, the pKa of that ionization, and pKa's will always be associated with a specific ionization, the pKa of that ionization is 6.37. So buffering will largely occur between 5.37 and 7.37 for this particular weak acid. Okay? If I'm below 6.37, I'm going to have more of the H2CO3 minus. If I'm above 6.37, I'm going to have more of the HCO3 minus. H2CO3 versus HCO3 minus, right? Okay. Now, you do some exercises, some of the problems I gave you, where you use Henderson-Hasselbalch equation to see relatively how much of those do you have at any given pH. And for a weak acid, Henderson-Hasselbalch is your best friend. Okay? I noted last time that maximum buffering capacity happens at the pKa. When the pH equals the pKa, buffer is at maximum buffering capacity, and the buffer is also at equal concentrations of acid and salt. All right? Very, very important concept. So when we have maximum equal concentrations of acid and salt, we will have maximum buffering capacity. Okay. Well, the Henderson-Hasselbalch equation for this ionization is shown here. pH equals pKa plus the log of the salt over the acid. Right? Remember that pH for acetic acid buffer would look like that. And in general, we can write it as the pH equals the pKa plus the log of the concentration of the salt divided by the concentration of the acid. Now, this tells us a couple of things. One is that there's only one constant in this equation. That constant is the pKa. The pKa is a constant for a given ionization. It's a constant. Now, notice I said for a given ionization, right? Because H2CO3 has two different ionizations that it can go through. It can lose the first proton and become HCO3 minus. It can lose the second proton and become CO3 minus minus. So I was talking only about the first ionization when I said the pKa value was 6.37. That ionization is the loss of the first proton. The second proton will have a different. The loss of the second proton will have a different pKa. When I say pKa, I want you to think two species, a salt and an acid, one that has one more proton than the other, and the difference being, of course, the loss of the proton during that process. By the way, I'm jabbering and I'm going fast. Nobody's asking me questions, so it's okay to ask me questions. I don't bite. All right? There's a question. There's a brave soul. Yes? Are non-protic acids best for buffers? Diprotic. Oh, I'm sorry. I was going to say non-protic. I'm not going to have too good of a buffer biologically for that. No. Any buffer is going to be best for whatever pH you want to use it at. So let's say I was interested in buffering something around 6.3, that H2CO3 minus H2CO3 and HCO3 minus would be a good buffer for that, but that would be a terrible buffer if I wanted to buffer something at pH 9. So there's no best or worst in terms of any given any just broad situation. It's a given situation, a given pH that I'm going to be interested in. So I use whatever buffer is appropriate for the pH that I want to buffer at. Make sense? Okay. Since I have to look all the way around the room, if I don't see you, just say, hey, Kevin. Okay. So strong acids, as I say here, like HCl, have no pKa value, essentially. All right? They completely ionize. We put them in water, we get all protons. We get all chloride. All right? There's no pKa associated with that. pKa values are valuable for telling us the relative strength of a weak acid. The lower the pKa value for a weak acid, the stronger that weak acid will be. The higher the pKa, the weaker the weak acid will be. I define everything in terms of being a weak acid or a salt. You notice I haven't used the word base, and I don't want you using the word base. We'll only use the word base to talk about strong bases like sodium hydroxide that completely dissociate. If you start using bases in association with these terms that I've talked about, you're going to get confused. So we don't. We talk about weak acids. Everything is a weak acid or a salt. The difference between a weak acid and a salt is a proton. Right? Very simple. Biochemists are simple people, like I said. Okay. Formic acid has a pKa of 3.75. Acetic acid has a pKa of 4.76. We would say formic acid is a stronger weak acid than acetic acid is. Because it will, if you do the math, and I think one of the problems I have, you do the math and you can see that in fact at a given pH, formic acid will have lost more protons than acetic acid will. It's a stronger acid. Okay, buffers, as I said, resist change in pH. They always involve weak acid systems. They have what we call buffering capacity. The greater the concentration of the buffer, the greater the capacity. We can exceed the capacity. One of the problems I used to assign students had a problem where they exceeded capacity of a buffer, and they got very confused because when they went to do that log term in the Henderson-Hasselbalch, they took a log of a negative number, and their calculator said, does not compute. And they came to me, and they always thought they did something wrong. And I said, you did it exactly right. Your calculator is trying to tell you something, but you don't want to listen to it. Listen to your calculator. It's telling you, if you can't calculate that, maybe there's something here I don't understand. And what had happened was they had subtracted more protons than they had protons available. They came up with negative protons. And much as we like to think about theoretical physics, I don't know anything about negative protons, right? They exceeded the capacity of their buffer, right? Remember that. When I say buffer, I mentioned last time, I want you to always think salt plus acid. It takes salt and an acid to make a buffer. If you have one, you don't have a buffer. Okay, so here's the example I just gave before. I won't go through that again. We see the rapid pH change at the low pH. We see the rapid pH change at the high pH, and you notice those rapid changes are happening outside of that pKa plus or minus 1. Limited pH change happening in the buffering region. Okay. Well, somebody asked about multiple protic. You asked about multiple protic acids. There are many multiple protic acids. We see that amino acids are always multiple protic. They always have more than one pKa value. Some have 3. Most have 2. And when we look at the titration plot of an amino acid, we see what you see on the screen here. Excuse me, I have a cold. We see 3 pKa values. This is an amino acid that has an R group, as we'll talk about in a bit, that can ionize. It has an amine group that can ionize. And it has an alpha carboxyl group, all of which can ionize. 3 different ionizations. If we look at a very low pH, we see that the predominant species that's there is the one that you see on the left. You'll notice as you look at it that it has all of the protons on. You look at the next one, okay, you look at about at about 1 equivalent, all right? That would happen probably at about pH 3, right? About 1 equivalent, you see one proton has come off, that very top proton, and the predominant species is the second one. The other protons are on. We go to adding of 2 equivalents, and we see that the second proton has come off, the second proton coming from the other carboxyl group. We go to a higher pH, we've added 3 equivalents, and we see that all the protons have come off. 1, 2, 3. Yes?
Can you clarify the definition of pKa versus Pi?
I'll talk about Pi in a second. Okay? So his question was the difference between pKa and Pi. So Pi is a special pH. It has nothing to do with pKa. Okay? Pi, since he's asked, I'll define it for you. Pi is the pH at which a molecule has a charge of exactly 0. Exactly 0. You can estimate where that is, but if you want to do the Pi, you have to calculate that. How do you calculate it? Well, it turns out to be pretty simple. Let's look at those species that we see above. We saw number 1 on the left that had all the protons on. What was the charge of that guy? What's the charge of the molecule on the far left? +1, right? What's the charge of the molecule to the right of that? And the charge of the molecule on the right of that? -1. And the molecule on the right of that? -2. Each one is a loss of 1 proton, right? So you could look at that graph and you could say, oh, well, I know the place where that molecule has a charge of 0. So first of all, I could estimate it by looking at the graph, right? Everybody could estimate the pH at which that Pi existed. You'd say, well, it's roughly between 2 and 4, right? To calculate it, it's very simple. You simply average the pKa values on either side of it. So when you do one of these, the first thing you do is you look and you say, where is the molecule have a charge of 0, and then what's the values of the pKa's on either side of it? You do that, you'll get the exact Pi. In this case, the pKa value for the one on the left is 2.1. The one on the right is 3.86. You average those, you've got the Pi. Notice, I didn't write that number out. I wrote the equation out. And that's what you're going to do. You are not going to crunch numbers in a calculator in this class. You are not going to have a calculator available to you to do that. All you're going to have to do is get the equation to the point of punching it into a calculator. You won't need a calculator. I'm not going to ask you what's the logarithm of 3.76, right? But if you got the thing down to the point where it said the log is equal to 3.76, then that would be punching it into a calculator, right? Okay. So what I've shown on the screen here now is what happens at the pKa value. At the pKa value, I have both species present. I have those 2 on the left present at equal concentrations at pKa1. At pKa2, I have those 2 middle ones abundantly and equally present. And at pKa3, I have both of those present. It's important to recognize what species is present at any given point on that curve. So I've shown you where we have 1. Now I've shown you where we have 2. You should be able to look at a graph and be able to interpret that. And you'll have some exercises in the recitation relating to that. Okay, so the rule I gave, this is one thing I won't give you on an exam, is something that you need to know. Estimating charge of a molecule. If the pH is greater than the pKa by 1 or more pH units, for the purpose of estimation, you may say the proton is off. Notice I said for the purpose of estimation. If the pH is less than the pKa by 1 or more units, the proton, you may assume, is on. So let's say I had that carbonic acid that we talked about before, and I gave you a pH of 3. And I said to you that the pKa values for the ionizations, one's at 6.37 and one's at about 9. What's the status of protons on that molecule? Are they both on, they both off, one on, one off? What do you think if I say pH 3? Everybody's frowning. Monday comes too early, doesn't it? pH is 3. Is pH less than or greater than the pKa? It's less than 6.37. It's less than 9. And it's less than by more than 1 unit, right? So, protons on or off? On. You apply the second rule, right? What if I said pH of 7.5? Come on, guys, wake up. 1 on, 1 off, right? Okay, so you can estimate this. I'm not going to give you that rule. You better know that rule. You're going to need to use it to estimate charge. Okay. You also need to know the relative charges. If a carboxyl group has the proton on, it has a charge of 0. If carboxyl has the proton off, it has a charge of -1. If an amine has an extra proton on, and it comes in various forms, it has the charge of 1. The amine has the proton off, it has a charge of 0. Those you'll need to know to estimate charge. And again, that's organic chemistry. Okay, that's the end of that lecture. We did the metabolic melody last time. So we'll stop that right there. Questions before I dive into amino acids? Shout them out. Okay, well, let's talk now up close and personal about amino acids. We're getting into structure function. Now, one of the things I'm going to tell you that you're going to like is you are not going to have to memorize the structure of 20 amino acids. Hey, some people make you do that. I think it's busy work. You do need to know, listen to this, you do need to know any amino acids that ionize and their charges as they ionize. That is, some of them have R groups that ionize, and those you need to know what charge that R group has when it ionizes. I'm not asking you to memorize the structure. Not going to happen. All right? I'm going to show you those as we go through. Let's take a look at them. Amino acids are the building blocks of proteins. You hear that, right? There are 20 amino acids that are commonly found in proteins, and 2 others I'm going to show you. Okay? Those of you that are nutrition majors have heard, and even if you're not nutrition majors, have heard about essential amino acids. What's an essential versus non-essential amino acid? I'll show you a list in a minute. Is that a question? Oh, okay. You're just waving at me. Hello there. It's a dead group this morning. You guys need a joke or something, right? No? Okay. We'll just go back to this then. You want a joke? Oh, yeah, we want a joke. Anything but biochemistry. I'm going to tell you my favorite joke. It's always the first joke I tell. But if you watch my videos, you've probably heard this joke before. Don't give it away. All right? This is Artie the Hitman. Have you heard of Artie the Hitman? Somebody said, yeah, they've seen the videos, right? Okay, Artie the Hitman. Little guy decides he wants to be a hitman. He wants to kill people, right? So he goes around the neighborhood. He puts up his little sign. And he says, we'll kill people for cheap, right? Puts his phone number down there and so forth. And this guy calls him up and says, hey, you're killing people for cheap? And he says, yeah. He says, how much? He says, I'll do it for $1. He said, $1? He said, yeah. He said, I'm trying to get started, make my name for myself, you know. And, okay. Who do you want me to kill? I want you to kill my wife. Okay. How do you want me to kill her? Oh, I get to pick that too? Yeah. He says, well, why don't you strangle her? Okay. Where's she at? So he said, well, she's down at the supermarket right now, you know. Describes her, and Artie goes down to the supermarket. And he sees the person in the description. He goes down there, and he looks around. There's nobody there. He goes up, and he strangles her right there in the supermarket. Oh, my God, right? He figured he's got it made. He's made that $1 now, right? He goes, oh, somebody saw him. Can't have a witness, right? Because you have a witness, you're a hitman, your career is over. So he goes over and he grabs the witness, and he strangles the witness, okay? He's about to leave, and he sees, another one. So he goes over and he grabs the third person. He strangles them right there in the supermarket. He decides, okay, I'm getting out of here. So he goes running out of the supermarket. The police go, and they catch him. And the headline in the newspaper the next day says, Artie chokes 3 for $1 at the grocery store. I ruined the punchline. Artie chokes 3 for $1 at the grocery. Okay. You like that one? It's better than if it were lettuce or something, right? So. Okay, let's talk about amino acids. After that, right? Basic structure of amino acids, all right? They all have a common structure, as we shall see. They all have, except for one, stereochemistry, meaning they have a left-handedness and a right-handedness. Only one does not have a left-handedness and a right-handedness. All of them are distinguished by their side chains, as we shall see, called the R groups that they possess. These R groups give each amino acid unique properties, and those unique properties, of course, in turn give the proteins that contain them unique properties. Ionization of these is important because ionization of amino acids causes charges to change. And when charges change, we see structural changes. Imagine a protein is all folded up, and one part of the protein up here has a positive charge, and it's right next to another part of the protein here that has a negative charge. We all know physics well enough to know they're going to attract each other. There's going to be a sort of attraction or bond that's going to happen with those. And if I change the pH such that the positive charge becomes 0, there's no longer going to be an attraction. And so we might expect that if we change the pH and we change the charge of a protein, we might change the structure of a protein, and in fact, that's exactly what happens. Okay? We'll talk a lot more about structure soon. Bonding, all essential things. Okay. Proteins are the workhorses of cells. Without proteins, we don't have cells. We don't have functioning cells. Proteins perform in catalysis, catalyzing reactions. Proteins perform in signaling, cells talking to each other in different parts of the body. Proteins give cells and the body structure. Hair is largely protein. Proteins are necessary for creating and storing energy. Proteins are comprised of 20 to 21, and I actually include 22 now, as I will show you. And they're broken into two groups from a nutritional perspective, the two groups being essential amino acids. And essential amino acids are those we have to have in our diet because we can't make them. No, you don't need to memorize this list. Okay? It's just there for your information. The list actually changes a bit. Different organisms, for example, have different essential versus non-essential. And even within organisms, like different humans, have different essential versus non-essential, particularly with respect to health and age. Some of these change in terms of some of them we can't quite make enough of when we're an infant, but we can as an adult. Okay? So it varies. But this is a pretty good list in terms of essential versus non-essential. All amino acids can be drawn essentially in the structure that you see on the screen. There's a central carbon atom that's there called the alpha carbon. That's found in every amino acid. Attached to the alpha carbon is an alpha carboxyl, and that's true in every amino acid. Attached also to the alpha carbon is the alpha amine, and that's also present in every amino acid. And last but not least, we have the R group, which is also present in every amino acid. The only differences is that one amino acid has a ring structure in which the R group is attached to the alpha amine. There's only one that has that. That's the amino acid known as proline, and we'll see that in just a little bit. There's an amino acid, an example amino acid, cysteine. We see that the central carbon, okay, is labeled, the alpha carbon, the alpha carboxyl, alpha amine, and R groups are all labeled, as you can see here. Notice that we have asymmetric centers. Every amino acid has at least one asymmetric center, and every amino acid except for I shouldn't say every one. Glycine is one exception that doesn't. Everything but glycine has an asymmetric alpha carbon. Glycine does not have an asymmetric alpha carbon because its R group is an H. And we see that H up there on the very top of the molecule. That's present also in every amino acid. So if we have two H groups, we don't have four different things attached to that alpha carbon. Okay, phenylalanine, another example. You can see exactly the same things here. Okay, I said handedness. We have stereoisomers of amino acids. Amino acids can be present in two forms. These are not amino acids, by the way. They're just two different forms of a molecule that has an asymmetric center, D-glyceraldehyde and L-glyceraldehyde, simply reflecting the fact that one carbon has 4 different molecules attached to it. And amino acids also are designated by D or L. We don't use the RS designation hardly at all in biochemistry. D versus L. Okay? Essentially, every amino acid that's found in proteins, whether it comes from a bacterium, a human being, a walrus, or a turnip, essentially every organism, folks, essentially every amino acid found in proteins is in the L configuration. Now, I said essentially. Whenever you hear that, you always think, there's got to be one catch, right? We'll talk about a couple of rare exceptions later in the course, right? But essentially, every amino acid found in proteins is in the L configuration. Why? Well, that L configuration is how life evolved. And that evolution of life, the three-dimensional configuration of the enzymes that made those amino acids only could make one form. Stereochemistry is very important when we go to think about putting things together. And enzymes are uniquely able to create one form versus another. If I take and make an amino acid in a test tube without an enzyme, I get random, D and L. When I look at amino acids found in meteorites that fall from space, I discover they are made chemically because they are random, D and L. We would love to find a meteorite coming from space that has everything in the L or the D configuration, because that would indicate it was not random and that some enzyme, in fact, had made that, which would indicate, wow, we just found evidence of life. We're still looking. Okay? All right, asymmetric carbons. All right, now, different books categorize amino acids in different ways. What you see on the screen is how I categorize them and how our book categorizes them. We have basically 6 groups. Nonpolar, some people call those aliphatic, and in some places I also call them aliphatic. Carboxyl, meaning that their R group has an ionizable carboxyl group. Amine, meaning that their R group contains an ionizable amine. Okay? Notice I said ionizable. Some things contain amines that don't ionize. Right? Aromatic, well, duh, they have a benzene ring within them. Hydroxyl, meaning that they contain a hydroxyl. And if you look at aromatic versus hydroxyl, you'll see tyrosine in both of them, and that's because tyrosine contains an aromatic ring, and attached to it is a hydroxyl. So it fits into both categories. And last, you see other. I'm going to break down other a little bit more for you later, but in terms of our categorization here, if you know these as other, you're fine. You should know the categories amino acids fit into. You should know the names of all the amino acids. You don't need to know the structures. The amino acids that have ionizable R groups are shown on the screen. Okay? Gesundheit. Aspartic acid, glutamic acid have carboxyls that can ionize. When they ionize, they form a COO-. The pKa values at which they do that are 4.1, and no, you're not going to need to memorize those. I'll give you that on an exam if you need it. Okay? Lysine, arginine, histidine have different amines that ionize, but you can see the forms that they have, and you can go back and look at their structures if you want. And different approximate pKa values for ionization. Of those 3, which one is the strongest weak acid? Of the 3, lysine, arginine, histidine. The strongest weak acid is the one that has the lowest pKa value. That would be histidine. Okay. Cysteine can also act as a weak acid. It has a sulfhydryl group that can ionize, losing a hydrogen and forming an S-. pKa value of 8.2, and we'll see that's a pretty important amino acid in the scheme of things. And last, tyrosine can lose an amino acid, can lose a proton. See, lose an amino acid doesn't make any sense. Lose a proton, become O-, pKa value about 10.5. Okay. Let's take a look at the structures that you're not going to need to memorize. You see why they're categorized the way they are. Here are the aromatics. The aromatics have an alpha carbon, they have an alpha amine, they have an alpha carboxyl, just like every other amino acid. They all have a benzene ring. At least one benzene ring in there. Tryptophan's the most complicated. Tyrosine and phenylalanine are related, the only difference being a hydroxyl group on the phenylalanine. That's what it looks like when they ionize at physiological pH. That's what they look like. You see the NH3 plus and the O minus. Right? Overall charge in these cases of 0. Okay? Notice that tyrosine at very high pH, remember the pKa value was 10.5 for the hydroxyl group, high pH value that will ionize. Here are the nonpolar ones. Alanine, glycine, proline. There's that proline I told you about. Proline has an R group that comes around and it attaches to the alpha amine. Proline. We're going to talk about a lot. Proline is interesting. It has less flexibility. You learned about rotation of bonds in general chemistry and in organic chemistry. It has less flexibility with its R group because the R group is physically tied to the alpha amine. Flexibility turns out to be pretty important consideration for proteins. If you have an amino acid within a protein, that section of the amino acid where that protein is located is going to be less flexible. There's going to be more constraints on the structure that it can have. One of the things we commonly see with proline in a protein is that where that proline is located, frequently we will see it's a bend in the protein. That bend reflecting that lack of flexibility. We see up here methionine on the lower right. Methionine is the only one up there that has a sulfur. You also saw a sulfur in cysteine. And in fact, cysteine and methionine are the only two that contain sulfur. Yes? Oh, that looks like it got cut off. You have good eyes. And that should have a hydrogen on there. Yeah. And when I ionize, you'll see, of course, the hydrogen's off. But yeah, thank you. I'll get that changed. Methionine and cysteine, only two with sulfur. Methionine is found as the first amino acid in almost every protein that's made on Earth. Almost every protein made on Earth. Okay? All right, most of these are pretty boring, though. Carbons and hydrogens in the R group. There's the R groups. There's the ionization. Not too surprisingly different from what you saw before. The alpha amine and the alpha carbon are the only things that are ionizing because the R groups don't ionize. Okay, there we go. Oh boy, how many of these do I have? Looky there, looky there, looky there. Okay, the amino acids with R groups that can ionize. Here's aspartic acid, here's glutamic acid. Okay, there's the R group. And you see a carboxyl out there, and you know carboxyls can ionize, and in fact they do. That's what happens to carboxyls at physiological pH. What's the charge on the aspartic acid at physiological pH? -1, right? Add all the charges up. Same thing for glutamic acid. Here's 3 amino acids that have hydroxyls in their side chain. They don't ionize with the exception of tyrosine. Serine and threonine don't effectively ionize at all for any practical purpose. Okay? You see the R groups. We see that serine is very similar to threonine. Okay? Tyrosine is somewhat different. Ionizes at high pH. You see that serine and threonine have charges of 0. What pH would it take to have tyrosine have a charge of -1? Anybody remember? What if I told you the pKa for tyrosine was 10.5? The R group. What pH would it take to have tyrosine have a charge of -1? So maybe brave and say aloud. 11.5. At least one pH unit above the pKa for the proton to come off. pH 11.5, tyrosine would have its proton essentially off. Okay, good. Cysteine. This guy ionized about 8. The number I gave you on the table before was 8.2. That's not too high above physiological pH. Cysteine turns out to be really important for protein structure. Because as we will see, cysteine not only ionizes, but cysteine forms covalent bonds very readily with the sulfurs of other cysteines. They form what's called a disulfide bond. So if I have a protein that has a structure up here that's got a cysteine where my finger is, and another part of the protein down here has a cysteine where this finger is, if they fold, which proteins do, and come into close proximity, what will happen is they'll go doink and form a disulfide bond. What's the strength of a disulfide bond compared to a hydrogen bond, folks? Much stronger. I have just now made a very strong link to help hold that protein together as a result of that. Cysteines play very important roles in stabilizing protein structure. We'll see some examples of that later. Okay? Here's two amino acids in the other category. And if you know them in the other category, you don't need to know what I have up here, which is that they're carboxamides. Notice they have amine groups. But I said that only those that had ionizable amine groups you had to know in terms of structure, in terms of charge, right? That amine on asparagine does not, on the R group of asparagine, does not ionize. Okay? It's a carboxamide. At physiological pH, you see no ionization of the R group. You only see the alpha carboxyl and the alpha amine, as you saw before. Asparagine and glutamine are derived from and related to aspartic acid and glutamic acid. The difference being they have an amine instead of an OH on that terminal carbon. Here are the amine-containing R group amino acids, arginine, lysine, and histidine. You can see their R groups there. And you can also see how they ionize at physiological pH. I tend to be lazy. I tend to just say amine plus charge. Because one of them has an NH plus charge, one of them has an NH3 plus charge, and one of them has an NH2 plus charge. And no, I don't care if you memorize those individual ones. If you know it's an amine that gets a plus charge in the R group, you're in good shape. What's the charge of these guys at physiological pH? +1. Yeah. Okay. Here's two odd amino acids. They're not coded for in the genetic code. We'll talk about the genetic code later. They get incorporated into proteins rarely and under unusual circumstances. We won't talk about that later. Selenocysteine is one we contain. We have probably about, I think, 50 proteins that contain at least one selenocysteine. Okay? Not a lot, but it's there. Has an important function. It's called selenocysteine because it looks just like cysteine, except for it has a selenium atom instead of a sulfur atom. A much more rare amino acid that's found in some proteins is called pyrolysine. It's found mostly in the archaean bacteria that make methane. I shouldn't say archaean. They're actually found in prokaryotic bacteria that make methane. Okay, there's alpha-amine. All right, all the various things there. There's that odd side chain on pyrolysine. It's related to lysine. In fact, it's actually made from lysine. And selenocysteine and all the blah, blah, blah there. Okay. There are some amino acids found in our body that aren't found in proteins. And no, you don't need to know the structures of those either. Carnitine is one. Okay. Others include citrulline, ornithine, and there are numerous others as well. So not all amino acids found in our cells are used to make proteins. Citrulline and ornithine, for example, are found as part of the urea cycle, the balancing of nitrogen in our bodies. Okay? Very, very important there, and we'll talk more about that later. All right, so let's go back now and look at that titration plot I showed you earlier for aspartic acid, and we can see what's happening during that. So this is important to kind of understand what's happening with the molecules. pKa1, we see the ionization from the one on the left to the second one from the left. That's happening at pKa1, or pKa1, same thing. And I would hope you would remember that they were equal concentration at that pH value of pKa1. The second pKa value we see right there. We would see these two being present in equal concentrations. And the third pKa value, we would see the last pair being found in equal concentrations. If we go outside of that range of the pKa values, more than 1 unit away, we would say, well, the most abundant species we have is the one on the left. We add up the charges, And why do we see charge says -1 when it should say a charge of +1? That's a good question, Kevin. I don't know. Kevin's going to change that one. Okay. Charge of 0, right? Pi value. There's -1, and there's -2. Yes, ma'am? Question over there. Yes. Thank you, by the way. Are the carboxyl groups always the first ones to ionize? You could learn it that way, I suppose, but I would rather have you learn it by knowing that the thing that has the lowest pKa value will always be the one that ionizes first. So if I gave you a sulfate, for example, sulfates tend to ionize lower pH values than carboxyls do. So knowing the pKa value in terms of understanding ionization would make more sense. Okay? But in general, carboxyls will ionize lower. Yeah. Yeah? By averaging the OH equivalents. If you average the OH equivalents, you would get 1. I'm sorry? Well, the average of the two equivalents of OH around the Pi there is 1.5 plus 0.5 equals 2 divided by 2 is 1. It wouldn't make any sense at all. The Pi is not 1. Well, yeah, it's always going to, well, no, that's not always going to take that. So you're going to look at different amino acids, and you're going to see some of them are going to happen at pK, that first ionization. Some's going to happen at the second. So that's why I said always look on the plot first. It's a good question. Always look at the plot first. Figure out where the charge is 0, and then do it accordingly. One of the things that students frequently do on exam is they think, oh, yeah, I've got to average the pKa value. So if I give them 4 pKa values, what do? They add them up and they average them. I can guarantee you that's not going to be right. All right? But if you lay this out and you see where the value is 0, then the 2 on either side you average. Does that make sense? Okay. Okay. How are we doing here? Time? Oh, we're doing good. Oh, question? Yeah. As I said, no. You are not responsible for knowing any of the pKa values. I will give any pKa values you need to know on an exam. Okay. Ionization changes charges. I showed earlier how that can happen. Here's 2 different proteins. We see their activity as a function of their pH, and you notice the activity changes. Maximum activity of pepsin is about pH 2. Where do we find pepsin? Pepsin is a protease found in our stomach where it's very acid conditions. It has evolved to be active at that pH. You get far out of that pH, pepsin's not active. Most proteins are active around physiological pH, and here's trypsin, most active around 6, a little bit higher later. But trypsin's also involved in digestion. There are post-translational modifications that happen to many amino acids. We'll talk about those more as we get going through things. But those post-translational modifications happen after the protein has been made. Hydroxyproline on the upper left there is one of the most common ones, and we'll talk about that very soon. Gamma-carboxyglutamate on the right, very important for blood clotting. Okay? Hydroxyproline is very important for strong collagen connective tissue. And many other things that can happen. You see the various modifications that happen there. Some of them are more elaborate than others are. Many amino acids can be phosphorylated, meaning attachment of a phosphate. And phosphate, by the way, folks, is negative at physiological pH. You attach a phosphate to serine, tyrosine, or threonine, you're going to make it from a 0 charge to a minus charge. Are you going to change the protein? You betcha. Learn that for the exam, right? Okay. There's a whole course we could teach on this. All right. Amino acid breakdown is very important. Amino acids are sources of energy if you're not getting much carbohydrates. You're on a low-carb diet, you're going to get energy from breakdown of proteins and amino acids. Some of that's shown right here. Okay. Now, I don't show this, put this on the screen and say, well, no, I've showed it to you. I can ask it to you on an exam, because that's not my purpose in showing it. I'm not going to ask you this on the exam. But I want you to see this because later in the class, next term, in fact, we're going to talk about that circle that you see there called the citric acid cycle. You can see how the breakdown of amino acids feeds the citric acid cycle. That's how we get energy from the breakdown of proteins. Okay. Wednesday, I'll talk about this right here, how we make peptide bonds. But in the meantime, we have something important. Please join me. Oh, don't do this to me now. Let's start that over. All right, we'll try that again. There we go. Okay, guys, you want extra credit? Lysine, arginine, and his basic ones you should not miss. Allyl, allyl, and met fill the aliphatic set. Proline, bens, and cis has S. Glycines, R is the smallest. Then there's terp and tyre and phi, structured aromatically. Asp and blue side chains of R say to protons au revoir. Glutamine, asparagine, bear carboxamide amines. Threonine and tiny ser have hydroxyl groups to share. These 20 amino A's can combine a zillion ways. Okay, guys, see you Wednesday.